So Otolith has many applications, and one of them increasingly recognized Otolith is the most reliable and stable and comparable structure for aging based on the basis of the daily, seasonal, and annual growth. So here's an example showing that we're using the three aging structures to compare the age determination of the liquid whitefish. So we use scale with the pectoral fin and the Otolith from the same fish give you the different aging results. So second one, these are three valuable fish species in the Great Slave Lake. Look at the whitefish, Incan lake trout. So bring you the different sort of Otolith shape. So Otolith can be used for as a reference for the species discrimination. Now today, I still say more about the importance of the liquid whitefish in Arctic lakes. The first in terms of the relative importance of the fish community study, and show you whitefish even dominated in the autumn, and even some other two the Coregonid still dominated in other water column, particularly in the surface, in the middle water. But I think the overall with whitefish is the #1 important species. By all means, I want to say the Great Slave Lake is one of the Coregonids dominated Arctic freshwater system. Liquid whitefish has sustained the largest commercial fisheries and the important subsistence fisheries in the Northwest Territory, Canada, since the middle of 1940. Here's some temporal trends of the commercial harvest before the 1972, particularly in the early 1950s. The annual harvest for whitefish can reach about 2,500 metric tons a year. And since 1972, the commercial fisheries statistic has been counted by aerial. So since the 1990s, commercial fisheries for whitefish have been downturning. Since the stock assessment result shows the population still is stable, but the market value has been a downturn. Third way, since the liquid whitefish is still as the pivotal functional groups in the Great Slave Lake ecosystem. So particularly connected to the lenses, from the bottom, and also be the part of the potential pre-resource for the high traffic level predator. So combined with the multi-species fish community study and harvest and ecosystem study, so liquid whitefish is most important in the Arctic Great Lakes. So the consequence question is to ask how many stocks of the liquid whitefish in the Great Slave Lake, and secondly, who is or are the most contributing to the area-specific or the basin-wide, fishery to the fisheries, and third, what are these stock status? By this, the statewide study, I can give you the whole bunch of the answers. We just using Otolith shape, and I give you the part of the answer. Now, since 2011, we designed the survey. Same thing here, just to show you that. And this is the main basin of the Great Slave Lake, and then eastern is this direction. So the Great Slave Lake is the largest lake in the world. And particularly in the eastern and the deepest area is over 600 meters deep. And because this is the main basin area, there are 6 administrative fish management area, and through this study we designed 245 grades. So we select a number of the grades based on the depth stratified mountain sample protocol. So this is the depth structure based on this map. They can show you the over 80% of the area are deeper than 30 meters deep. So during the study, we collect whitefish is the number one species. So through the study, you can see the 3 different type of body shape. So particularly like the main 2 factors you can observe easily is the color and the body shape. No, this from the deeper area, and this from the outer Ocean River and the Snake River, and since here is the River, since that's the western basin. So most of the deeper area in the Yellow Bay area. So in this study, we included 4 groups of the variables here, including the environmental condition using the Y-side reading. Also, is the general biological observation with the lens weight and single. And most importantly, we use the R-based shape R to generate the Otolith mathematical parameters such as the Otolith area, length, parameter, width. And based on this measurement, we generate the 6 Otolith-shaped indices. And lastly, we also propose the 7th Otolith-shaped indice. We call it the Otolith density, based on the Otolith weight divided area. So this just gives you the general background, the temperature example. It shows that in the summertime, and the surface of the Great Slave Lake, it seems like the uniform structure on the surface about 18 degrees, but there is evidence the soil stratification in about 10 to 12 meters deep. So here is in the bottom area, things that are deeper than 10 meters, all around 5 degrees down in the whole bottom area. Now here is the survey results of whitefish and abundance and biomass density. So you can see that the whitefish can appear everywhere, and most of the large size of the whitefish appear into the nearshore area for feeding. Okay. So now we did 2 trial studies. The first one we compared the pairs of the Otolith on the left side and the right side, because sometimes we just suddenly broken the one or missed the one. So we will see if we use the other one as for Otolith shape analysis. So this study showing here seems like the significant difference between the left side, because different direction, just like in my hand, like this. If you flip overlap, may be the same, but here just overlap is different. So statistical result showing significant difference. But since based on the deviation things that of the outline, things that at where the 5 levels, things that the deviation of the outline is under the 2%. So here is the example. Here the red line just generated by R-based shipped on soft package. Now, this is the data things to compare to the left and the right. So things by aerial, by obvious aerial length. So and the other portion of the table that has the male or the female. So the result of the non-parameter analysis showing no difference of the length in the left and the right side of the orbit based on measurement of the length, width, and aerial parameter. And significant is found between sets. So the second trial study, we just stick to the left side of the orbit from the 500 Whitefish. We use the polar coordinate system defined from the 0, the posterior into also 90 degree and 180 and 270. So that's based on this direction. We compare to the other Whitefish orders from the different area. And so this result showing similar to CV. So the big difference thing you can find is the one is in the posterior area. You see the very wide variation, and second in the anterior. So based on the 500 left side of the others, and so we can all combine all the data, others indices and the measurement. So based this from using a hierarchical cluster analysis, define a shape phi ecotype we call it. Now this one just plot area based. So the fisheries management area based the composition of the star. So it's an ecotype. So things that in the western basin is the one west and one east. Things that just the 3 stars in one west and the 3 in the area 3. And the same is the area, so it's 1. And because in the summertime, all ecotype overlap in the same area because the different stock of the fish they move into the shallow area for feeding. But the same is that type E, particularly it only appeared in one east and area 4. So that's a deep water depth. So based on this study, since the 5 ecotype study, so you can see the ecotype A showing based on this table about the figure. So here you see the big AR and the lowest the CL. So compare to the LE. So compare with the big Otolith and the bigger is the CL, bigger ID. ID like the roundness. So you can look at the Otolith shape. Since ecotype A, since like the elongate, and the ecotype B, since like a rounded, close to the round, and the ecotype C because the low density. So the Otolith things pretty thin. And so the ecotype D or E here is the small Otolith but very thick. So I think the density is the highest. And the ecotype D since like the transitional, so the between and the A, B, C or E. So prior to using like RDA, prior to the all measurement of the Otolith and Otolith the industry, and combined with like the environmental data. So things are 3 important environmental variables combined with the change of the Otolith shape. So first like conductivity, so conductivity, temperature, and the result of the oxygen seems to be the environmental variable to control the shape of the otolith. And second, the overlap of all the ecotype of the group of the fish over like the environmental layer together. So you can say that the ABC can be defined in the upper area since like this is a low oxygen and this area be high oxygen. So this is ABC. And since the B, since like you perform to like clean water scenario, but ecotype B because the deep water. So the conclusion from this spectral-wide study is that we find that no significant difference exists in the left and the right sides of otolith shape, but differ by sex, size, and spatial distribution. Second, 5 ecotypes of the liquid whitefish were identified, and most of which were overlapped during summertime, some feeding time period. But ecotype E seems to be proposed in the deeper water area, characterized by great otolith density. And over 6 environmental variable, conductivity, oxygen, and temperature have been greatly impacted formation and spatial distribution of the 5 type of whitefish in Great Slave Lake. Now I like to see the acknowledgement for funding source and involved of Aboriginal community participating in the summer survey, and still have many summer students through the co-op and the festival to join the study. Thank you.
If you try to calibrate with fish age, did you age the fish?
Yeah.
Try to calibrate to save the age.
Yeah, we aged each of the otoliths, but in this study we did not include it, just simply to choose the random sample. In fact, we still notice some correlation to the age structure.
Well, it's not quite clear to me how you can make inferences on the density of whitefishes from the area. Because the area might be the same, but some of them might just be thicker in the third dimension, so they have the same density then. Should it be based on the volume instead of the area?
That's a good point. Thank you. Yeah, things that, well, first, fisheries management, whitefish, commercial fisheries in the Great Slave are based on the area. So the depth is a very important factor. Even here, since I use the RDA showing the depth is not dominant factor. I show you here the RDA plot. So depth is not dominant factor, influence the composition of the ecotypes.
I didn't mean the depth of the environment of the lake, but the height of the otolith.
I see. Height of the otolith. Yeah, we did not do it because R is the shape only to do like the outline for each otolith.
Right.
Yeah, that's a good point.
So you're missing the height then?
Yeah, good point. Thank you.
You have measured the left and right side. You haven't many analyses on integrating measurement. It's also possible, but they're so identical left and right side. Many times there exists differences in left and right side.
just like a statewide study, the following work still continued. Since first future plan, first, we need to increase the sample size. Second, we need a sample like a spawning stocks. Because in the feeding, summertime, the feeding period, all stocks just overlap. So that gives us a certificate just based on the subjective study. Now we're back to the sample. We need to get like some mature spawning fish and be able to give us based on like spawning facilities. So the thing that we need a spawning stock sample. And third, we need to combine like a genetic study. So genetic study and a body shape analysis, Otolith shape analysis. Body shape analysis as an external body shape. And Otolith is like a stable internal pattern. So three aspects of the study combined together, then we can get the conclusion how many stocks at last exist in the free stable data.
Quick one?
Probably not, but I'll just flag it for Xinhua. I just was curious about your Xinhua, where is the cutoff to make the equal-types going down through your dendrograms? It just wasn't clear to me when I was looking at that. It sort of looked like there were being more clusters than 5. Anyway, we can talk about it later.
Yeah,
the same.
Yeah, that's a good point. Depending on the cluster analysis, you can choose, different people use a different criteria. Maybe we will sort out the different number of the stars. And then nowadays, just assume based on the minimum the distance criteria. So we divide 5, maybe 4 or 6, maybe.
Yeah.
So
you have to kind of choose a point where you feel that it makes sense, I guess.
yeah, exactly.
Okay, thank you very much. Okay, our last speaker is Timothy Parks.
Is it alright if I just hold this? Alright. I won't be into too much. I promise. Where I can't get it. So. I'm just afraid of losing the microphone, so I'm just gonna do this. So we started this study in 2011, and it was pretty much a part of a larger study where we looked at the overall status of inland Cisco resources that we have in Wisconsin. And I'd like to acknowledge all the folks that are on this author list, as well as others who've been a part of this study as well, particularly Dr. James Church, who had a large part in taking the digitized photos and analyzing the body morphometrics that we first looked at. So what we know about Wisconsin's inland populations before 2011 was fairly limited. And as we started this, we started to really begin to understand how important these populations really were. And so this is really much more of like the initial portions of like figuring out what these populations really mean in their ecology. So we're still trying to put a lot of stuff into the picture as to what things mean and how that impacts fisheries, as specifically game fish fisheries. But we do know some things, such as it's an incredible ecological asset in many different ways, including it being a high-energy prey source for many of our game fish species and having a large influence on growth dynamics of these species, as Dr. Greg Sass has just mentioned yesterday in his speech. Also, because of its sensitivities to particular environmental perturbations, it's a pretty good indicator species as well as we look at these lakes and we look at water quality and such. Because of its capacity to structure zooplankton communities and its capacity to perform middle-out trophic cascades, potentially, it's this ecological engineer as well. So it's this pretty unique species that we're just trying to get a handle on. And as we looked at these populations, we figured that these populations were fairly diverse in many sorts of ways as explained through population dynamics, but just obviously as you look at the body forms of these fishes, these things are very, very different within Coregonid Symposium as we look at them. And we think, like, as we're trying to figure out their ecology and how these specific forms came about and such, we have all these questions about, like, what do these forms mean and how can we use this towards resource management and fisheries-related applications. And such things that we think about as do certain body forms influence gear selectivity. Like if you have two fish of the exact same size, but one's a little bit thicker, is it gonna be vulnerable to the same size net mesh if you're having a vertical gill net? Or does it have the same acoustic size characteristics when you look at hydroacoustics and you're trying to analyze the single echo detection? So I think about these things. We also think about things particularly like, do larger or deeper bodied cisco, do they impact the availability of certain prey to, or them as prey to specific predators with possible gait limitations, as that might be some sort of a bioenergetic bottleneck. As we saw yesterday with Greg Sass, that even in some of those lakes that he had, it really wasn't a large measurable effect where cisco were present. So maybe part of it could be the fact that some of these cisco might not actually be there and available to be eaten. And lastly, there's been a lot of anecdotal information and personal observation from other folks such as Pete Jacobson, noticing that some of these morphotypes that we have out there have clear associations with water quality status, but not a lot has been really looked towards looking at some of these allopatric populations and how they reflect current lake conditions, such as these deep-bodied cisco, also known as tulibes locally. They tend to be pretty commonly occurring in these lakes that would have marginal habitat, where you'd have low dissolved oxygen and large proportions of hypolimnion, and also really warm epilimnion, and so you'd have this squeezed habitat, and so we tend to find these grotesque yet beautiful tulibes, which, I mean, when I pulled this fish up, I was like, oh my gosh, is that some kind of a gizzard shad? Is that a freshwater drum? I was like, no, that was an RTD. That was cool. Anyways, but there is a large body of knowledge that we currently know about morphs and morphometric variation, and it's fairly obvious throughout the northern hemisphere, inter and intraspecies wise, and it's both seen allopatric and through sympatric populations within different systems. And there's much debate over how a lot of this phenotypic variation can be explained. You know, a lot of it seems to be characterized by environmental or ecological explanations, but also much of it can be, or there's some theorized that could be potentially due to genetic variation, but not, you know, a lot has been really done there, so that's an argument I really can't get into, and it really doesn't have much to do with my project yet so far. But our basic charge was to look at the morphological variation across Wisconsin's lakes and see what we see and describe what's there. Really try to figure out a little bit about the ecology and like what sorts of habitat associations or lake characteristics might help explain some of this variation that we see. And then, is there any sort of way that we can help biologists describe this, because they might not have the tools that we have or the extreme knowledge of multivariate statistics that we have. So is there any other surrogate measurements that they could use to help describe some of these morphs as it may help them to manage their resources? So as it started out in 2011 through 2014, we had this large synoptic status assessment of ciscos throughout Wisconsin, which was 133 lakes where we found these fish species, where we performed vertical gill net sampling. And as you see here, we placed these vertical gill nets. They 7 different panel meshes going from the top of the water column all the way to the bottom, and we placed those in the deepest portions of our lakes, and we basically distribute or allocate our effort based off of the complex bathymetry that we'd have in these legs. And this is pretty much the preliminary work that we've done. And from this work that we saw, where we were able to find fish, we were able to find 17 populations of fish that had at least 30 individuals for us to perform this analysis. It represented a range of different habitat and lake characteristics that would tell us stuff about what's happening there. A wide spatial distribution as well, and this pretty much got us started for what we were trying to look at and such. And so with each of these samples, we obtained our individuals that we needed, took all the specific fisheries data that we need as well, and then retained these fish specimens as best as we could for frozen transport back in the lab, where we performed these digital photos where eventually they'd be used to create this grid system where we place these 15 homologous landmarks in a simplistic fashion at common insertion points that are used in other Cisco species, and then we'd be able to perform geometric morphometric analysis. And just the goal of it really essentially was just to try to get a handle on what was out there and such. And once we were able to correct for allometry or size, we were able to see this, where basically there's this gradient we were seeing which had a lot to do with body depth. And we were seeing these deeper bodied or terete-shaped fishes going from this edge, the upper left-hand corner, which would be Bear Lake, all the way to the bottom right-hand corner of this relative warp analysis, or PCA, which would be Geneva, Lake Geneva, which will come in later to describe this. It can be easily broken down again using cluster analysis to really these deep body forms and these shallow body forms from these lakes that we analyze in just a very simplistic way. There's obviously some more specific attributes that can be looked at as well, but for simplicity, we're really seeing this body depth gradient. Just getting a handle on like what could be associated with this, not necessarily how these fish are responding to these things and having these morphological variants, but what these things were associated with. We looked at a range of different factors, including spatial factors, bathymetry, aspects of water quality such as oxythermal habitat, TDO3, which is a temperature at dissolved oxygen of 3 milligrams per liter, which was described by Tyler Arenstorff, and TSI to describe water quality patterns as well. But we found out through all this that TDO3, this temperature at dissolved oxygen 3 milligrams per liter, describing oxythermal habitat. And essentially there was this positive relationship where the higher the temperature you'd see with this oxythermal habitat, the larger or the deeper body fish you'd come across, which is pretty easily described here through this gradient of isotherms. And as you see, going up in temperature all the way up to Bear Lake, which would be described right here, you'd see these larger or deeper bodied forms. And down here you'd see these more ovate or shallow body forms as with Lake Geneva. And just here's an example of how much habitat Bear Lake would have, which is probably about 1 meter or less of functional habitat with a pretty warm temperature, versus Lake Geneva, which has a lot more habitat and a fairly cold temperature where you'd see 3 milligrams per liter. So now that we had like a handle on like, oh, this is kind of what we'd expect to see, is there some sort of a measure that could really help us with describing these morphs in a way that could help managers essentially look for them? And so we thought about it for a while. We thought about relative weight, or basically a way to measure condition to show that, well, here's the difference between this deep body and the shallow body form. It would be an easy way to do it. And since we had this classification using cluster analysis, we decided to create a logistic regression, which came out to be pretty accurate. It's about 93% correct classification rate of describing the probability of having a deep body form, and the 50th percent probability of going across, and having this deeper body form, this threshold would be at around a relative weight of 90. It may differ depending on our sample size and such, but from what we're seeing in various randomizations of the data, it seemed like right around 90 was where you have this threshold for having these two of these. And taking that information for what we had actually increased our sample size of geometric, morphometric information, or just morphometric information. So we're able to use a larger number of populations to really kind of look at what sorts of things are reflecting in this oxythermal habitat. And it seems to me that right here, if we look at the deeper body morphs versus the shallow body morphs, we're seeing that a higher temperature at a dissolved oxygen of 3 milligrams per liter being represented by these deeper body fish versus the shallow body morphs being more in these environments that have sometimes more habitat, but definitely colder habitat as far as quality would go. Just as a summary for all this, we just really wanted to have more of a simplistic presentation, so I would go fast and we knew this is the end of the day, so everybody's probably groggy and such. We just want to do this really quick, but what we learned is that there's a great diversity of populations out there of Cisco within Wisconsin, but the majority seem to be highly reflected in these two predominant body forms if we look at things in an allopatric sense. And somewhat it seems to be associated with this oxythermal habitat dynamic and this gradient if we look across lakes. Body forms too were easily identified once we looked at this through this relative weight index, which was really easy to do. We're really not sure what's the mechanism behind this, and we could really kind of dig into the weeds as to what's happening. It could be trophic, it could be genetic. A lot remains unanswered as to what's actually happening, which could be the start for a lot of folks to start questioning. Now that we can identify these, what's the matter? How did they get there? Was this always there? Was this plasticity, rapid evolution? There's all these questions that can emerge. But we do have this relationship with TDO3, and it's really interesting. And there's I think it's a pretty good potential for another water quality indicator. And there's a lot of information out there that's looked at sympatric populations where there's habitat segregation, and it's largely based on temperature. But also in the future, I think there's lots of work that can be done with these morphs, and specifically genetics, life history traits, and then also broad population rate dynamic functions such as production or growth that could be done. Because I think there's a lot more that's poorly understood just about these that can really greatly clarify what we're looking at with these populations. And with that, I just want to thank everybody who's been involved with the project and everybody's help with the field data collection, folks from Wisconsin DNR, UW Systems, specifically Stevens Point, and this nice lady who contributed a lot of our artwork, so that was a big help. So thank you.
Your results seem counterintuitive. Coregonids are cold water species. Why should fish that are restricted to warm water habitat be equine fish? I wonder if it's just due to lake productivity. The lakes that have poor oxygen concentrations in the deeper water are more likely to be more productive.
That's kind of what I was thinking as well. There's some sort of a trophic response, oh, these things are coming like this because they're changing essentially how they consume food. Is that what you're thinking, or? Yeah. All right.
Did you look at the productivity of this very soon?
Only indirectly through these measures of TSI, or I guess oxythermal habitat would be some sort of a surrogate, but not a true. I mean, a lot of the information like that, like total phosphorus, was kind of unavailable, so it was difficult.
Yeah.
Okay.
I was just going to say, in Minnesota, from what I presented yesterday, we're kind of seeing similar trends as you're seeing, Tim. And I was curious from the trophic state index things as well. you know, we're seeing a lot of relationships between like phosphorus and Cisco size, and maybe you guys don't have good phosphorus data, but I think that's something that, you know, could be helpful as well in describing why you have these different morphs. I guess another different question, but related, is, you know, you kind of talked about the shallow and deep body morph, and you were kind of, you know, describing them as being sort of independent of each other. But, you know, do you really see these two different morphs, or is it more of a gradient that you would see in Cisco between maybe a shallow and a deep body? But did you see more of this gradient along the way or not?
I'd say, yeah, there was not like a clear, like, you know, distinction between them, but there was definitely a gradient. If you go back to that PCA, it was, you know, you could just take the relative weight measures as they were highly redundant with those PCA loadings, and just essentially just, you know, measure how it responded to those direct measurements. Like, you're exactly right. And so it wasn't like just a clear thing that, all right, this is very discrete from each other, but that's a great question. Oh, or
yeah, just go back on the traffic suggestion that you did on this as a mechanism behind it. Did you check about thermal developmental component? Because it's being shown that thermal and developmental component will affect the body's shape and disease, that would work really well. So it would be an added taking those.
yeah. That's actually something that John Lyons suggested early on as having like a particular influence to some of these fish, and it's something that would have to be answered at a later time when we can actually start doing controlled experiments for these things. But yeah, no, that could be a largely you know, explanatory to what we're seeing. One more.
Just one more here.
Yes.
One thing is body height of Coregonid, very big differences. Variability just in that type of the high
Can you speak a little bit louder, sir?
temperature and oxygen. One can ask how much they can impact if you think you're going to be feeding the food.
Oh, like their
It's a
metabolism?
very important component in this environment, and this can also be the reason that we will also grow much better. And if your schedule, say, will be similar, very, very keen and small.
Yeah, no,
Feeding is a very important thing
to consider as far as that. Yeah, I think you're absolutely right. I think that's probably a lot of the case too.
very much.
Like, as it influences their metabolism and their growth, their somatic growth, yes.
But then we have also the genetic background and also real being behind this. So it's really things perhaps to take account.
Yeah, there's
Temperature and oxygen.
yeah, there's a lot to consider as far as that goes, especially the genetics, because we're really unsure if there is genetic structure there or not. So, yeah, that's very good stuff to think about. Thank you.
Okay, thank you very much. So thanks to all the speakers, including Julia. Session 4 o'clock today. And Jerry will run down what's happening.
Is it going to be posted? Is that going to be posted? Awesome.
Okay, so first off, if you are a Northland College alum, which is the college just down the road, if you could meet me at the side here, just after this, after the logistics for tonight. The tour to NEDF is going to begin leaving at the business center at 5:15. We're going to treat those shuttles a lot like we did last night with the buses. So as that as that van fills, that will take off, and the next one will take off, and we'll have to do 2 loops. So we want to get everybody up there by 6. So please be to the business center by 5:15 or shortly thereafter. The vessel tour is much more casual, starts at 6, and there will be crew members on each of the boats, and the hack, and the kayak are already here, and another boat is on its way in. And feel free to mingle down the dock. There will be some pretzels and bottled water if you need a light snack before you head off to dinner. But that tour is much more casual, so take advantage of it. Anybody have any questions about this evening? Tom?
You say the vessel tours, does it start until 6?
At 6, yes.
Are we going to be at the aquaculture day at 6?
Well, the folks, there's a limited number of people that can go to the aquaculture center, and you had to register for that trip because they can only handle a smaller size group. But other folks it'll be a kind of a nice alternate. Any other questions? Great day today. Thanks for all the speakers.